Hello everyone, welcome to ITLS Academy. Today's lecture is about the structure of nucleic acids. Nucleic acids are long chain polymers of nucleotides which are joined by the phosphodiester bond. A nucleic acid is consist of sugars, nitrogenous bases and phosphate groups. When sugars and nitrogenous bases are joined together, then it is known as nucleoside, and when phosphate group is added to the nucleoside, then it is known as nucleotide. Nucleic acids are the polynucleotides which is formed in a chain by phosphodiester linkage. Now here we will discuss about the different type of sugars in nucleic acid. Generally there is a two type of sugars found on a nucleic acid. One is ribose, another one is deoxyribose. The major difference between a ribose and a deoxyribose sugar is that a ribose sugar contains a 2' hydroxyl group, where the deoxyribose sugar lack the free 2' hydroxyl group. Generally, the deoxyribose sugar is found on DNA and ribose sugar is found on RNA. Now we will discuss about the different type of nitrogenous bases that are found on the nucleic acid. Now here you can see that the basic structure of purine is consist of a single benzene ring that is accompanied by another ring, but in case of pyrimidine there is only a single benzene like ring. Okay, now here just see the numbering pattern, that is anti-clockwise in purine, but in pyrimidine it is clockwise. Okay, now from that you can easily remember the basic structure of different purines and pyrimidines. Now just see, adenine is 6-aminopurine. 6-aminopurine mean it has a amino group in its 6th position or 6th carbon here. Okay, now guanine is 2-amino 6-oxypurine. Now look, it has a amino group in 2nd carbon and it has an oxy group in the 6th carbon. Okay, now cytosine is 2-oxy 4-amino pyrimidine. Now here have a look, that is 4-amino here and that is 2-oxy. That is the structure of cytosine. Now thymine is 2,4-oxy 5-methyl pyrimidine. Now have a look, 4-oxy, 2-oxy and 5-methyl, that is thymine. And uracil is 2,4-oxy pyrimidine. Now there is a difference between uracil and thymine, that is the thymine contains a 5' methyl group, but in uracil there is a lack of 5' methyl group. Uracil is generally found on RNA and thymine is found on DNA. Now here we will discuss about the bonding pattern between the sugars, nitrogenous bases and phosphate group. Now have a look. When the sugar is attached to the nitrogenous bases, that is the C-N bond, that is known as the glycosidic linkage, and a phosphodiester bond forms when the sugar attaches with a phosphate group. Now have a look, that is a ribose sugar and it is a ribose sugar. This is the phosphate group. This is the phosphate group. Okay, now this OH and this OH interact with each other. As a result, water is removed and a bond is formed. This bond is known as phosphodiester bond. So phosphodiester bond is COPOC. That is this funda, or that is the easiest way to remember a phosphodiester bond, COPOC. Okay, now here we can discuss about the RNA hydrolysis. We all know that DNA is a very stable structure, but RNA is not a stable structure. Now we will know about why DNA is so stable and RNA is not so stable. The reason is that the RNA or the ribose sugar have a 2' hydroxyl group, but the deoxyribose sugar lacks it. Under alkaline condition, the 2' OH group is deprotonated and it works as a nucleophile which attacks the phosphodiester bond. When it attacks the phosphodiester bond, it cleaves and it creates a 2', 3' nucleotide monophosphate. The cleavage is done here. Okay, this is the 2', 3' nucleotide monophosphate and another part is cleaved. So that's the reason. Another part is cleaved here. So that's the reason that RNA is not so stable and DNA is stable. So thank you everyone for listening to our lecture. Keep visiting our website. Thank you.